Hello. In today's video, we shall learn in detail about the anatomy of both dicot stem and monocot stem. By the end of this video, you'll have a clear understanding about the different zones seen in the transverse section of the stems and also about the types and arrangements of vascular bundles along with their differences. Let us start with the introduction to angiosperms. Angiosperms are flowering plants and bear seeds. They can be divided into two main groups, namely dicotyledons and monocotyledons, based primarily on the number of cotyledons. Those plants whose seed contains two cotyledons are called dicotyledons or dicots, while those plants whose seed contains only one cotyledon or embryonic leaf is known as monocotyledons or monocots. As we all know, the stem supports the plant, facilitates the transport of water and nutrients to different parts of the plant, and also contains special tissues that help in the growth of the plant. Let us look into the anatomy of a dicot stem, example sunflower, and find out the different zones present in it, and also study in particular about the type and arrangement of vascular bundles. The internal structure of a dicot stem, example stem of sunflower, mainly consists of epidermis, hypodermis, cortex, endodermis, pericycle, vascular bundle, and pith. Epidermis. It is the outermost layer consisting of a single layer of cells. The cells are thin-walled, flattened parenchymatous cells arranged compactly without any intercellular spaces. The outer walls of the epidermal cells have a thick cutinized layer called cuticle. The cells of the epidermis do not have chlorophyll, but some cells are modified to form multicellular hairs or trichomes. Epidermis shows the presence of a few stomata. The thick cuticle minimizes the rate of transpiration. The epidermis protects the underlying tissue from mechanical injury and also against different environmental factors. It helps in exchange of gases through stomata. The epidermis of dicot stem might get replaced during secondary growth. The cortex is the zone that lies in between the epidermis and the pericycle. It is composed of three distinct regions, namely external hypodermis, general cortex, and internal endodermis. The layer immediately below the epidermis is the hypodermis, which is several layers thick and is composed of collenchyma cells. These cells have thickenings at the corners made up of cellulose and pectin. Collenchyma cells are living and contain a number of chloroplasts and therefore help the plant in preparing food. The cells of hypodermis provide mechanical strength, elasticity, and additional support to the epidermis. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Next is the general cortex. It lies below the hypodermis. The general cortex region is composed of few layers of thin-walled, rounded or oval parenchymatous cells arranged loosely with adequate intercellular spaces. The cells of this region store the food prepared by the cells of the hypodermis. Here and there, the general cortex shows the presence of resin canals or mucilage canals. Endodermis, it is single-layered and is the innermost layer of cortex consisting of barrel-shaped cells which are compactly arranged without any intercellular spaces. The endodermis separates cortex from the vascular bundle. It stores a large amount of starch grains and is also known as starch sheath. The radial and transverse walls of the cells of endodermis are thickened due to the deposition of lignin, resulting in the formation of the Casparian strips. Pericycle. This is the region lying in between the endodermis and the vascular bundle. Pericycle is made up of patches of sclerenchymatous cells above the vascular bundles. It not only gives mechanical support to the plant body, but also protects the underlying tissues. It also has intervening masses of parenchyma which serves to store food. The cells of the pericycle are sclerenchymatous and thus it is often called as hard bast. Vascular bundle. It is inert to the pericycle and is composed of xylem, cambium and phloem. They are arranged in the form of a ring around a central pith. The vascular bundles are wedge-shaped, conjoint, collateral and open. The vascular bundles are limited in number and have a uniform size. In a dicot stem, xylem and phloem combine into one bundle, xylem being internal, phloem external with cambium in between them. Xylem lies towards the pith. It consists of tracheids, vessels, xylem parenchyma and xylem fibers. The tracheids and vessels are composed of smaller protoxylem and larger metaxylem. The protoxylem lie towards the center, while the metaxylem lie towards the periphery, and hence the xylem is said to be endark. Xylem fibers are thick-walled, lignified cells surrounding the metaxylem. Xylem parenchyma are thin-walled living cells surrounding the protoxylem. The function of xylem is conduction of sap. Phloem. The phloem is present below the sclerenchymatous patch of the pericycle and occur in the form of undifferentiated mass. The phloem consists of sieve tubes, companion cells, and phloem parenchyma. Companion cell is always found associated with the sieve tube. All the above elements are living, thin-walled, made up of cellulose. Phloem helps to conduct food materials from the leaves to other parts of the plant. Cambium are a group of meristematic cells lying between the xylem and phloem. These cells are living with large nucleus and dense cytoplasm. Cambium are capable of repeated cell division. They give rise to secondary xylem and secondary phloem and therefore bring about secondary growth resulting in the thickness of the plant body. Pith. Pith is well defined in dicot stems. It occupies the central portion below the vascular bundle and is made up of rounded or polygonal, thin-walled, living parenchymatous cells with intercellular spaces between them. Cells of pith store food and help in the conduction of food and water between the bundles. Medullary rays, they are thin-walled, radially elongated, polygonal parenchyma cells present in between two vascular bundles. Now, we shall look into the transverse section of monocot stem. The internal structure of a typical monocot stem, example, stem of maize, consists of epidermis, hypodermis, ground tissue, and vascular bundles. Epidermis. Monocot stems are protected by an outer layer of dermal tissue called the epidermis. It is made up of single layer of tightly packed parenchymatous cells with thick cuticle. Multicellular hairs or trichomes are absent, but may show the presence of a few stomata. Hypodermis. A few layers of sclerenchymatous cells lying below the epidermis constitute the hypodermis. It gives mechanical strength to the plant. Ground tissue. It is not differentiated into cortex, endodermis, pericycle, and pith. Ground tissue is a continuous mass of loosely arranged, thin-walled parenchyma cells extending from the sclerenchyma of the hypodermis to the center. The cells of ground tissue enclose numerous prominent intercellular spaces. It helps in the storage of food. Vascular bundles. In monocot stem, the vascular bundles are found scattered in the ground tissue. They are numerous in number. Near the periphery, the vascular bundles are small and lie very close to each other. Near the center, the bundles are larger in size and are widely spaced. A bundle sheath made up of sclerenchyma cells is present which surrounds the vascular bundles. The vascular bundles are oval in outline. They are conjoined, collateral, endarc, and closed. The vascular bundle consists of xylem and phloem, but cambium is absent. Monocot stems lack secondary growth due to the absence of cambium in their internal tissue system. If they show increase in girth, as seen in palm trees, it does not result in the formation of secondary xylem and secondary phloem since vascular cambium is absent. An increase in girth without secondary growth is referred to as anomalous thickening. Xylem consists of vessels, tracheids, xylem parenchyma, and limited xylem fibers. The vessels are arranged in the shape of the letter Y, with larger two round pitted metaxylem vessels forming the arms and smaller annular or spiral protoxylem vessels forming the base. Xylem are endarc, with outer metaxylem and inner protoxylem. Fewer tracheids are present between the metaxylem vessels. Thin-walled wood or xylem parenchyma surrounds a conspicuous cystolysigenous water cavity which stores water. This cavity is the result of breakdown of the inner protoxylem vessel and parenchyma during the rapid growth of the stem. Phloem In monocot stem, the phloem consists of sieve tubes and companion cells. Phloem parenchyma is absent. The outermost portion of the phloem, which is a broken mass, is the protophloem, and the inner portion is the functional metaphloem with distinct sieve tubes and companion cells. Pith is not differentiated in monocot stem. Now, let us study the differences between dicot stem and monocot stem. In dicot stem, the hypodermis is made up of collenchyma cells. In monocot stem, the hypodermis is made up of sclerenchyma cells. Epidermal hair or trichomes are present in dicot stem. Trichomes are absent in monocot stem. In dicot stem, the general cortex is made up of few layers of parenchyma. In monocot stem, general cortex is a continuous mass of parenchyma extending from the sclerenchyma to the center. Stele in dicot stem is differentiated into pericycle, vascular bundle, and medullary rays. Stele in monocot stem consists of ground tissue and vascular bundle. Vascular bundles in dicot stem are arranged in the form of rings. Vascular bundles in monocot stem are scattered throughout the ground tissue. In dicot stem, vascular bundles are fewer in number and uniform in size. In monocot stem, there are numerous vascular bundles of different sizes. Smaller bundles are towards the periphery, while the larger ones are in the center. In dicot stem, the vascular bundles are collateral and open. In monocot stem, the vascular bundles are collateral and closed. Bundle sheath are absent in dicot stem. Vascular bundles are covered with sclerenchymatous sheath in monocot stem. Pericycle, medullary rays, and pith are present in dicot stem. Pericycle, medullary rays, and pith are absent in monocot stem. In dicot stem, cambium is present and the stem exhibits secondary growth. In monocot stem, cambium is absent. There is no secondary growth. Phloem parenchyma are present in dicot stem. Phloem parenchyma is absent in monocot stem. So today we have learned in detail about the internal structure of dicot stem and monocot stem and also differences between the two. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.